Good morning, my dear students. A couple of days back, we were discussing about water relations. I was just telling you the importance of the relation of water with the plants and animals. You know, the plants and animals, they depend on water for their life activities. So how the plants are able to absorb the water, this is what we were discussing in our earlier class. Now, in water relations, another very important topic is what is called as transpiration. It is about this we are going to discuss in our class. So, our topic is going to be transpiration for today. First, let's try to define the word transpiration. What's called a transpiration? It is the evaporation of water from the aerial organs of the plant. I repeat, it's the evaporation of water from the aerial organs of the plant. Okay, right. Now, why we are using the word evaporation? There is a loss of water from the plant even in the liquid form. Now, when the water is lost from the plant in the liquid form, it goes by another term, what is called as guttation. It is called as guttation. Now, what is this guttation? Guttation is the loss of water in the liquid form. You could have seen, if you are in the habit of visiting your garden in the early morning. I'm sorry, I can't expect this from the children of this age group. You can't go in the early morning during, particularly in this season, to your garden. But if you have got that a good habit, you could have seen that at the end of the grass leaves, in the margin of the grass leaves, you could have seen the water droplets directly coming out. Many of you mistake this for what is called as a snowdrops. No, it's not a snowdrops. Actually, water is exuding out of the plant in the liquid form. So, when the water comes out of the plant in the liquid form, it is called as a guttation. And when it is escaping from the plant in the form of the water vapor, you call it as transpiration. Now, there are different types of transpirations are there. One is called as a stomatal transpiration. Second one is a cuticular transpiration. Third one is lenticular transpiration. You know, stomatal transpiration or stomata are the organs, small organs, we are going to discuss about the stomata in a very detailed way. So, the stomata are very small organs, and when the transpiration is taking place through the stomata, you call it as a stomatal transpiration. Since stomata are present on the surface of the leaves, you call it as a laminar transpiration. Is another word for that. You know, lamina means leaf. Lamina means leaf. So it is also called as laminar transpiration. Some people call it as foliar transpiration also, because once again the word folia means leaf. Folia refers to the paper, folia refers to the page, folia refers to the leaf also. So, foliar transpiration is another name for stomatal transpiration because stomata are present on the surface of the leaves. Okay, fine. Then, cuticular transpiration. What is this? On the surface of the leaves, you will be able to see a structure, a wax-like coating. And this wax-like coating is called as a cuticle. Now, cuticle is, to some extent, it is preventing the loss of water. It is preventing the transpiration. But anyhow, some amount of transpiration is taking place through the cuticle also. So when transpiration is taking place with the help of the cuticle, we call it as a cuticular transpiration. Okay, fine. Then lenticular transpiration. What is this lenticular transpiration? These structures are called lenticels. They are a little bit big opening. They are nearly hundreds times larger than the stomata. These lenticels are present on the surface of the bark. So when the water is escaping in the form of water vapor through the lenticels, you call it as lenticular transpiration. Okay, so now till this time what I have discussed, I am just going to give you a quick recap. The escape of water in the form of water vapor is called as a transpiration. The escape of water in the form of the liquid is called as a guttation. And this transpiration we divide into three, namely stomatal transpiration, cuticular transpiration and lenticular transpiration. Of these three types of respirations, stomatal transpiration is taking place in the plant to the maximum amount. Nearly 90 to 95% of the transpiration that is taking place in the plant is of a stomatal type. Why it is like that? Because stomata are uniformly present on the surface of the leaf, and then you have got a huge amount of stomata on the surface. How much of the stomata are present, how the transpiration is taking place through the stomata. These are all some going to be very interesting things in biology, and that's what we are going to proceed with the next slide. Transpiration. It is estimated that a tomato plant loses 155 liters of water in one growing season. Just to see. And then one corn plant loses 245 liters of water in one growing season. The loss of water from a forest in USA is about 36,400 liters per acre, and you see it is a per day it is. Then according to Huber, only 0.1% of water is lost by the bark, that is lenticular transpiration. I was telling you just now that the majority of the transpiration is of the stomatal type, stomatal transpiration. So stomatal transpiration and cuticular transpiration, they form the maximum, and the lenticular transpiration that is taking place through the bark is of very insignificant amount. Okay, fine. So as I was just telling you, stomatal transpiration is a very important transpiration. That's going to be our area of main discussion today. So stomatal transpiration is taking place through the stem and leaf epidermis. And these two organs, the stem and the leaf, are provided with numerous pores called stomata. Of course, I have just stated that stem and the leaf epidermis are provided with numerous pores, but they are present everywhere in the plant. They are present on the leaves, they are present on the stem. You know, they are present in the flowers also. If you just take the petals, the sepals, and do the anatomy of this, you will see that the stomata are present in the sepals and petals also. Don't get astonished if they are present in the pistils also. They are present everywhere except the root. Even on the fruit wall you are able to see the stomata. But these stomata are very thick, very small, and they don't do that much of a transpiration. So from our point of view, from the physiology point of view, from the transpiration point of view, stomatal transpiration taking place through the stem and the leaf is more important, even though stomata are present on the other organs, namely flower, fruit, etc. Okay, now the next point is the number of stomata per square centimeter of the leaf epidermis is variable. It is highly variable. There are certain leaves which are possessing very little number of stomata, and there are leaves which are possessing huge number of stomata. Here I have given normally the stomata present in 1 square centimeters, but normally in most of the textbooks they would have given the number of the stomata present in every square millimeter, because in 1 square millimeter, you know, millimeter, it's a very insignificant area. When I just put a dot here, just a dot, that dot will be more than 1 square millimeter. That small dot will be more than 1 square millimeter. Now in that small dot, the area which I am just touching with the tip of the pin, in that area nearly 200 to 300 stomata will be there. That's why when I am telling that the leaves are provided with a number of stomata, most of you may be asking, sir, you are telling that the leaves are possessing the stomata, but we are not able to see. Fine, your question is a good question, but you see, you can't see the stomata with your naked eye. The stomata are so small that it needs microscopes. Even simple microscopes you can't see the stomata. Only with the help of your compound microscope you will be able to see the stomata. Even in a compound microscope, in one focal field you will be able to see about 80 to 90 stomata will be there. Such small microscopic structures are the stomata. Okay, so in 1 square millimeter you get a good amount of them. And you see, I have given the data here for every square centimeter. The lower epidermis contains 10,000 stomata in Z maze. You know what's a Z maze? It's a cholum. We call it as a cholum in our language. And 39,000 in Pyrus malis. I have given everything in binomials. What's a Pyrus malis? Pyrus malis is a very simple plant. You must be knowing that. But since I have given the name in binomial, you are not able, may not be able to understand. But it is a simply apple. Pyrus malis is apple. Pyrus communis is pears. So in apple, you are getting 39,000 stomata per square centimeter, and it is 58,000 in Quercus retilina. So this gives you an estimate of the number of the stomata present in the different surface of the leaves. Now you know, see, as I was just telling you, then second point, if you see the number of the stomata per square centimeter of the leaf epidermis is variable, here you will be able to see that statement highly correct with the data here. It is ranging from 10,000 to roughly 60,000. So much of variability is there. Now you may be asking, why is such an amount of variability is there in the distribution of the stomata? Now it depends upon the nature of the plant, the place where the plants are living. When the plants are living in a very dry area, perhaps the number of the stomata will be very less. You must be knowing that to reduce the amount of transpiration, the number of the stomata are getting reduced. So in xerophytic plants, when the plants are living in a dried climatic conditions, you get the number of stomata very less, and in the water plants, hydrophytes and mesophytes, the number of the stomata will be naturally more. So there are so many parameters which determine how many stomata should be present or distributed on the surface of the leaf, but to that aspect we will not go now. We are more interested in knowing about the method of transpiration taking place through the stomata. So we will go to the next slide. See, from now onwards we are going to learn about the structure of the stomata. A stomata consists of two guard cells which are generally bean shaped or kidney shaped. Then in grasses they are dumbbell shaped. The guard cells measure 45 to 15 mu in size. Okay, now I want to give some explanation regarding this slide. A slide consists of two God cells which are generally bean shaped. Okay, now I am just going to focus a different type of a stomata on the next slide, but for the explanation here I am just going to draw a single stomata. See, these two are called as the God cells, and surrounding these God cells you will be able to see a number of cells. These are called as accessory cells, or it's also sometimes called as subsidiary cells. Accessory cells and subsidiary cells. These are the God cells, and this is the stomatal pore. You see, the God cells are kidney shaped in structure. See, I just tell you one very interesting thing. One day a zoology teacher came and then he was explaining to you about the germination of the bean seed. He thought that the children may not be knowing about the bean seed, and then he was just telling the bean seeds are in the shape of your kidney. Then what happened? The botany teacher came and then he was explaining about the bean seeds. He thought that the children may not be knowing about the bean. He told that bean seeds are kidney shaped. As a result of that, the students didn't understand the structure of the kidney as well as the structure of the bean. Anyhow, now I have drawn the shape on the board, so you must be able to understand what is the shape of the God cells. So this shape we call as a kidney shape. The God cells are kidney shaped, and then they are surrounded by a number of accessory cells, leaving a small pore in the center called a stomatal pore. Now, in this circumstance, I want to clarify certain things in your mind. Some anatomists, they call this structure as stoma. They use it as in the singular form. They call this as a stoma, and when many stoma are present, they call it as a stomata. According to them, stoma is singular, stomata is a plural. Okay, now we understand that? Fine. But some anatomists are different, and they say that this stoma refers only to the hole, only to the pore. Only to the pore it is called as stoma. And the other region, they are using a different term called stomatal complex. Stomatal complex. Those anatomists who are using the word stoma for this full structure, they call this as a stomatal pore. So, a little bit of a controversy is there in the usage of the term of stomata. But anyhow, for your level, you just understand this as a stomata. This as a stomata. Okay. And it is used in a singular form as well as in the plural form. So, a stomata is surrounded by a number of accessory cells. In grasses, these God cells, these are dumbbell shaped. They are dumbbell shaped. The God cells measure 45 to 15 mu in size. Fine. I don't know how many are able to understand this term mu. See, these are all what are called as a sub-multiplication table in biology. You have to understand these measurements very clearly, particularly in anatomy and cytology. When we go, very often we will be using the term mu, micron, etc. So, those students who don't know till now exactly what a mu is, I just tell you, you make a note of that. It's very, very important. When 1 millimeter is divided by 1000, you call it as a micron, and when it is a return in this form and pronounced as mu. One thing I want to clarify, mu and micron, they are one and the same. It is a return as a mu and pronounced as micron. So when 1 millimeter is divided by 1000, it is called as 1 mu or 1 micron. You know, the meter is the basic unit of length in physics. So when 1 meter is divided by 1000, it is a millimeter. When still divided by 1000, it becomes a micron. So micron is represented by 1 into 10 to the power of minus 6, because we'll be getting 6 zeros. So micron is expressed by that value. So 1 into 10 to the power of minus 6 meters. That's very, very important. It is 1 into 10 to the power of minus 6 meters. Now when 1 micron is still divided by 1000, then it is called as a nanometer. Then it becomes 1 into 10 to the power of minus 9. Nano means 9, you must be knowing. Okay, it's also called as a millimicron. So meter, millimeter, micron, nanometer. It's like that. Fine. Now you must be please be thorough with this table, because we will be using these words very often in anatomy and cytology, particularly the terms micron, millimicron, nanometer, etc. Now once again I am telling you, the God cells measure 45 to 15 mu in size. That means now you must be able to visualize very clearly what will be the size of 45 micron. That is 1 millimeter divided by 1000 and then 45 units. If you are taking that much smallness is the size of the God cells. Now these God cells, they enclose a tiny opening called stomatal pore or stoma, or it is also called as stomate. The diameter of the stoma or the stomatal aperture is 5.4 mu in dolichos, and it is a 27.5 mu in avena. As the size of the stomata is variable, as the distribution of the stomata is highly variable. Now we will see that the dia of the stomatal aperture is also highly variable. It is a 5.4 mu in dolichos. What is a dolichos? Dolichos lab is beans. So very simple. Dolichos lab, full name is Dolichos lab. Dolichos lab is the another name for beans. You can note down. Then avena. What is avena? Avena sativa is the botanical term for oats. Oats is a very common plant. Every one of you must be knowing about oats. So when I say oats, you understand it. But when I say avena, you are not able to understand. But avena is the botanical term for oats. Okay. So in avena, the stomatal pore is a 27.5, and it is in the case of beans, it is a 5.4. It's also highly variable. That means stomata will be having a very, very small hole in the center, or the hole may be very big in size. Okay. The God cells are surrounded by subsidiary cells, or what are called as accessory cells. Now, depending on the type of the accessory cells, the stomata are divided into different types. Very important types alone I am going to just now tell you. See, these are all the God cells, and if you have two cells, two subsidiary cells parallel to the God cell, then this is called as parasitic stomata, because this is parallel to this. This is a parasitic stomata. Okay, right. Now this type of stomata is what is called as diacytic stomata. See, this is diagonal to the God cells. These are diagonal to the God cells. I am writing it so you can note down. Parasitic stomata. This is diacytic stomata. Diacytic stomata. Now, when a pair of God cell or when God cells are surrounded by two large cells, two large subsidiary cells and one small cell like this. See, these two are large cells and this is a very small cell. This is called as anisocytic stomata. Anisocytic stomata. You know, in biology, aniso means dissimilar, iso means similar, aniso means dissimilar. So here three subsidiary cells are there which are dissimilar in size. So you call it as anisocytic stomata. Now, when you have got a pair of God cells surrounded by three equal cells like this, three cells are there which are equal in size, then it is called as anemocytic. Anemocytic. See, these are the very fundamental type of stomata which was recognized by the anatomist about 100 years back. It's a really an astonishing fact that the study of stomata has so much grown and developed that they have nearly recognized 120 types of stomata in literature today. See, starting from 4 types of stomata, today we have got nearly 120 types of stomata the cytologist and the anatomist have recognized. We are not going into the details of that because our main concern is about only transpiration, which is concerned with the stomata. If you are going to the cytology, a separate area where we will be studying about the stomatology as a different structure, then we will discuss more about this. Okay, fine. So we will stop with this about the structure of the stomata, and now we will go to the other aspect of transpiration. The inner tangential wall of the God cells are thickened. The cellulose microfibrils of the God cells are radially arranged. I just draw the diagram. I will be able to show this in the slide also, perhaps in the next slide. This wall is very thick. This wall is very thick, and this wall will be thin. This wall will be very thin, and this wall will be extremely thicker. So at least 7 or 8 times thicker than this. So it is because of this thickness. It is formed of microfibrils, and because of the shortening and elongation of these microfibrils, the stomata are able to open and close. When the stomata become turgid in nature, they become open, they remain open. And when the stomata becomes flaccid, the opposite term for turgidity is flaccidity. When they become, they become flaccid, that means the stomata will become more or less like this, you see, and the stomata pore will be very small, very small. This is a flaccid stomata, this is a turgid stomata. The stomata will remain like this during the daytime, and it will remain like this during the night time. So during the night time they become flaccid, during the daytime they become turgid. When they remain turgid, they remain open. When they become flaccid, they remain closed. Understand this very clearly. Now, because next slide I am going to explain to you how this opening and closing of the stomata is taking place. Very interesting. The nature has given a power to the plants that the stomata normally they remain open during the daytime and they remain closed during the night time. You need not to close or open the stomata one by one. It's not possible. They automatically remain open during the daytime and they close during the night time. Nature has got some amount of what is called as a mechanism for this. What mechanism is operating for this opening and closing of the stomata is what I am going to explain to you in the next few slides. And so many theories have been forwarded by the different cytologists and the physiologists, how this opening and closing of the stomata is taking place automatically in the plant. It's all by chemistry. It's all by chemistry. Different concentration, osmotic pressure, and pH of the solution. These are all playing a very vital role in the opening and closing of the stomata. See, even though I told that the stomata or opening and closing of the stomata, to that aspect we will go a little bit after this slide. Based on the distribution of the stomata, you have got different types. They are namely amphistomatic. The leaves are amphistomatic. Some leaves are hypostomatic, and some leaves are epistomatic. You know, amphi means two or both. You must be knowing about it. See, amphibians is a name of a class, including the frog, toads, etc. After Pisces, amphi means two. That which is able to live on the water as well as on the land, you call it amphibia, something. So, amphistomatic, when the stomata are present on both on the upper epidermis and lower epidermis of a leaf, then that is called as amphistomatic. Good example of this category is oats. Then hypostomatic. Hypo means below. So when the stomata are restricted to the lower epidermis, you call it as the leaf as a hypostomatic. The good examples are apple and mulberry. You call a leaf as an epistomatic when the stomata are present only on the upper surface. See, in some cases the stomata are present on both the surfaces. In some leaves it is present only on the lower surface, and in some plant it is present only on the upper surface. So based on that, you divide the leaf into apistomatic, hypostomatic, and epistomatic. I have given the examples for each category here. Now, coming to the movement of the stomata, I told you, normally the stomata, they remain open during the daytime and they close during the night time. That's what I told you. Now, you may be raising a basic doubt, why the stomata should remain open during the daytime and they should close in the night time. This is a basic doubt for any student for that matter. Now, during the daytime only, different processes, the different life activities like photosynthesis, respiration, all these activities are taking place mostly during the daytime because the plants are very active during the daytime. And so, the stomata, they should remain open to absorb the, they absorb the oxygen and then they give out the carbon dioxide during the respiration. They take in carbon dioxide and give out oxygen during the photosynthesis. For all these purposes, the stomata should remain open during the daytime. So, the transpiration is also taking place very actively during the daytime, and normally they remain closed during the night time. I am using my words very cautiously because there are so many exceptions to this particular general rule. There are certain plants in which stomata remain closed during the daytime, and there are many cases where the stomata remain open during the night time also. See, whenever there is a rule, there will be always exceptions in biology. The rules are accompanied with exceptions. Okay, fine. So, in these are all the exceptional cases that I am giving to you. In a plant called alfalfa or lucerne, in a plant called alfalfa or lucerne, they remain open during the daytime and they close during the night time. This is a normality. So, the good examples are pisum. You know pisum plant? This is a pea plant. Lathyrus. Lathyrus is a sweet bee. Then brassica is a mustard. Then cucumis. The cucumis is a plant belonging to the family Cucurbitaceae. Then helianthus, a sunflower. In all these cases, the stomata remain open during the daytime and they close during the night time. Then potato type. In potato, what happens? They remain open throughout the day and night, except for a few hours in the evening. See, only for a few hours in the evening they close, but they remain open throughout the day, during the day as well as during the night. The examples of this category is cucurbita. You must be knowing about cucurbita. That is cucurbits, coming under family. Cucurbita is a pumpkin, the best example. Allium. Allium is what? What is allium? Allium is an onion. Onion and the garlic are called as allium. Allium. Cepa is onion. Allium. Sativum is garlic. Okay. So in allium and cucurbita, they open mostly except for few hours. Okay. Then the last type is what's called as a barley type. They open only for a few hours during the day and never open at the night time. So the good examples are cereals. So what are the cereals? Cereals we call as a dhaniyangal. In Tamil we call it a dhaniyangal. See, all the plants coming under the Poaceae family, the last family in your taxonomy, they are all called as a cereals. Rice, wheat, sugarcane, oats, barley, millets, these are all the group called as cereals. Just like a pulses, parupugal, they are all coming under the family Fabaceae. The cereals are coming under the family Poaceae. Now, the stomata of Eucysitum is a pteridophyte. In English, it is called as a horse tail, and in a plant called Cypus validus, they remain continuously open. There is no opening and there is no closing of stomata in these two plants. Now we are coming to what is called as the mechanism of opening of the closing of the stomata. So there are so many theories about this. Photosynthetic production within God's cells is the first theory. Now what this theory states, during the photosynthetic activity is responsible for the opening and closing of the stomata. This idea was given by a great scientist called Von Mohl in 1856, long back. So he says that photosynthetic products are very, very important in the opening and closing of the stomata. Some more theories are going to be presented to you. In that you will understand that that theory is only an elaboration of this theory. We will go to the second theory, permeability hypothesis. What is called a permeability hypothesis? Carbon dioxide. When carbon dioxide is depleted from a plant, it is more or less like a photosynthetic activity only. See, the carbon dioxide will be depleted or it will be utilized only during the photosynthesis. When a photosynthesis is taking place, the plant will utilize the carbon dioxide and then it will be liberating the oxygen. So this depletion of the carbon dioxide from the plant is the most responsible thing for the closing and opening of the stomata. That's what they say. The third theory is what is called as a starch-sugar interconversion hypothesis. As I was just telling you, this third theory is only an elaboration of the first theory. During the photosynthesis, what happens? Carbohydrate, starch is formed, sugar is formed, it's converted into starch. Then this is I'm sorry. During the daytime, due to the photosynthetic activity, starch is produced in the plant. You know, the God cells, they contain a good amount of chloroplast, and these chloroplast. Now the God cells, when compared to the accessory cells, they contain nearly 3 to 4 times number of chloroplast will be more. So more chloroplast means more starch will be produced by the God cells. Now these God cells will produce more starch, and then that will be converted into sugar, and this results in the God cells become turgid, and this is responsible for the opening of the stomata. What is taking place in the night? It is just the reverse. It's just the reverse. The sugar is once again converted into starch, and that leads to the flaccidity of the God cells, which results in the closing of the stomata. Now you understand why during the night time the stomata remain closed, and why during the daytime it remains open. So it is the starch which is being produced. During the daytime, the chloroplasts are able to produce a starch which are converted into sugar, and that is once again responsible for the closing and opening of the stomata. Some more pictures I am going to depict to you. So now you can understand this very easily with the help of the pictures. Now you see is a diagram of a turgid stomata, and here this is the stomatal pore. Then see the wall. This is how the walls are very thick when compared to this wall. See? The wall surrounding the aperture is very thick. Here you see this is the diagram of a flaccid stomata. During the night time they remain closed. See? Okay. So stomata, singular stoma, pores in the leaf mostly on the undersurface. Each pore is surrounded by a pair of God cells. God cells can change shape to open or close the stomata. This is how a open stomata remain, and this is how a closed stomata remain. Okay. The same diagram in the transverse section. These are the God cells, stomatal pore, subsidiary cells, surrounding cells, or called as accessory cells. So this is the same thing, but here you see this stomata remains open and this stomata remains closed. It's due to the movement of water getting through the osmosis. It's going out during the by the diffusion. See, God cells, chloroplast, epidermal cells. Of course, the God cells are not, chloroplasts are not seen very clearly. These are all the chloroplasts. They are shown green color. When compared to this cell, more number of chloroplasts will be there. More number of chloroplasts. I was also telling you 3 to 4 times the number of chloroplasts will be more. Water diffuses into the God cells, which causes them to open. On hot or dry days, the God cells have less water. They relax, and then the stomata close. Mechanism of opening and closing of the stomata. The fourth theory states that it is due to the potassium transport hypothesis. This was presented by a scientist called Imamura. He told that potassium ions, when they go into the God cells, they are responsible for the opening and closing of the stomata. And this gained prominence more because they were able to show that more amount of potassium ions when the stomata are open, they were able to record the potassium ions to the maximum possible extent, and the potassium ions to the minimum level when they are closed. So the amount of potassium ions present in the God cells, the way in which they are transported to the God cells, that is mostly responsible for the opening and the closing of the stomata. See, this diagram clearly explains to you about the activity of the potassium ions for the closing and opening of the God cells. God cells take up potassium ions, and it results in the active transport. That is what is happening for the opening, and the reverse is for the closing of the stomata. So when the potassium ions are pumped out of the cell, when they are pumped out, they become closed. When they go into the God cells, they remain open. So it is the potassium ions which are really responsible for the opening and closing of the stomata. The last theory about the mechanism, which explains about the mechanism of a theory, is called a proton transport hypothesis, proposed by a scientist called Leavitt in 1924. Now it is a combination of Karth and Stefan's hypothesis and gave a modified version of the stomatal movement. Now what he was telling is about the potassium pump. I was just explaining to you. Now he says the protons and potassium both are equally responsible for the opening and closing of the stomata. This is what proton transport hypothesis is telling. Under the influence of light, protons formed by the dissociation of a malic acid move from the cytoplasm into the chloroplast of the God cells. Then to counter the exit of protons, potassium ions enter God cells from the surrounding cells. So what I want you to understand is the protons are responsible, and by the dissociation they form the malic acid which is responsible for the opening and the closing of the stomata. That's about the last theory. Now we have reached mostly the final aspect of the transpiration. Now which are all the factors responsible for the stomatal movement? I am just going to explain. There are 6 factors are there which are responsible for affecting the stomatal movement. First one is light. During light, stomata remain open, and during darkness they close. This I have explained to you many times sufficiently in my class today. However, the stomata of ecosystem remain open throughout the conditions. This point also we have discussed. So light is responsible for the stomatal movement. Second one, the stomata do not open under ultraviolet or far red exposure. Best stomatal opening occurs under red and blue wavelength of light, but they fail to open under green wavelength. So now you understand the effect of light on the stomatal movement. The next one is carbon dioxide concentration. This I was explaining to you while I was explaining the mechanism of a stomatal opening and closing. Due to high carbon dioxide concentration in the intercellular space, the stomata close even during the daytime. So when there is a depletion of carbon dioxide concentration, it leads to the opening. I was telling you. So when the carbon dioxide concentration is increasing, it results in the stomatal closure. So carbon dioxide concentration is very important in the opening and closing of the stomata. Third factor is pH concentration, pH of the God cells. This I was explaining to you by the 5th theory. So pH also plays an important role, and increased pH favors the opening of the stomata, whereas low pH favors the closure. Understand that very clearly. Increased pH favors opening and low pH favors the closure. Okay. Then the next factor which is affecting the stomatal movement is temperature. If the temperature is below 0 degrees Celsius, the stomata remain closed. So even in the daytime, see, even during the daytime, where the stomata are expected to remain open, but if the temperature is below, then the stomata will remain closed. So light factor is different, temperature is different. Even in the presence of light, if the temperature is below 0 degrees Celsius, then the stomata will remain only closed. Now we understand the role of temperature in deciding whether the stomata should remain closed or open. Now the last factor about the stomatal movement is the abscisic acid. What is an abscisic acid? An acid which is responsible for the leaf fall. See what happens is very interesting in biology. This is a stem and you have got a leaf here. See what happens when an abscisic acid will be produced at this area before the leaf fall. See, when the leaf is going to fall, the abscisic acid will be produced, then the leaf will fall, and then that will help in the wounding of the heel in the plant. Now, this abscisic acid is responsible for the transpiration also. When transpiration rate exceeds absorption, the plant is subjected to water stress. As a result, incipient wilting occurs. Due to loss of water contents, the stomata are closed in several mesophytes. Now, the transpiration is affected by many factors. These are divided into two categories, namely environmental factors and plant factors. I will just rush through this. It's very easy to understand these factors. First, we will go to the environmental factors and then we will go to the plant factors. Now, if you understand that the transpiration is more or less like an evaporation, then memorizing these points become very, very easy for you. See, transpiration, I told you, is an evaporation of water. So, it is more or less like an evaporation. When humidity is more, transpiration will be very less. You know, see, when there is a humid atmosphere is there, even if you put the cloth for drying, it will not get dried. Because humidity is more in the water droplets are more in the atmosphere. Then, how it will get dried? So, humidity, more humidity means less transpiration. Less humidity means more transpiration. It is in the, it is conversely proportional. Okay, inversely proportional. Temperature. When temperature is more, transpiration will be more. When temperature is less, transpiration will be less. It is directly proportional. Similarly, light also. When more light is there, more transpiration will be there. During the night time, there won't be any transpiration. Darkness leads to the closure, light leads to the opening. Wind is a very complex process. See, when there is a mild wind is there, a slight breeze is there, that is promoting the transpiration. But when there is a heavy blow of wind is there, then the transpiration rate will be reduced. When there is a mild blow of wind in the form of a breeze, then the transpiration will be more. Okay. Then atmospheric pressure, it is reverse. When more pressure is there, transpiration will be very less. When less atmospheric pressure is there, transpiration will be more. That is how atmospheric pressure is acting as a very important factor affecting the transpiration. Then last one is availability of a soil water. When more soil water is there, evaporation will be more or a transpiration will be more. When there is no sufficient amount of water in the soil, then of course the transpiration will also be very, very less. Then factors affecting transpiration, plant factors are there. Now first factor is the root and shoot ratio. Now when the root is very less, the shoot also will be very less. When the root is very strong and extensively branched, very deep root system is there for a plant, then the shoot system will also be more. So there is always what is called as the root and shoot ratio for us. Now this root and shoot ratio is deciding the amount of transpiration in the plant. So it is this factor which is very important for the transpiration. When more shoot and root shoot ratio is there, more transpiration will be there. Root shoot ratio is very, very less transpiration will be there. Similarly, leaf area, when leaf area is more, you will have the more transpiration. You will know in some of the xerophytic plants, when we are going to the environmental studies, I will teach you more about how the leaf area is determining the transpiration. In some of the plants which are living in a desert area where there is a hectic sunlight is there, the plants do not have any leaf at all. It's all completely reduced to the thorns and spines. Why it is like this? It is simply, it is a mechanism to reduce the leaf area. So when the leaf area is very less, transpiration will also be less. Then the structure of the leaf is very, very important. When the leaf is having a cluster of hairs on its surface, more cuticle is there, more wax coating is there, sometimes the leaves will fall from the plant. So all these things are leading to what is called as a less amount of a transpiration. So the some of the plant factors affecting the transpiration is root shoot ratio, leaf area, and the structure of the leaf. Now, this is what is called as a Ganong's photometer, which is used to measure the rate of transpiration. A bubble will be introduced from here. When the plant is transpiring, the bubble will move through this capillary tube. Then you will be able to exactly understand how much of a transpiration has taken place. First, since it was designed by Ganong, you call it as a Ganong's photometer. The same photometer is a different design. That's all. When the plant is transpiring, water will get reduced from here. How much of water that the plant has taken, you can very easily calculate because it is a calibrated pipette. This is what is called as a farmer's photometer. It's all different. Design only different. It was designed by a scientist called Farmer. So you call it as a farmer's photometer. When this plant is transpiring, the amount of water used by the plant you will be able to calculate, and then you will be able to, in a unit time, in a plant with this much surface leaf area is able to transpire this much water. All these things you can easily calculate, but the calculation part of that I am not discussing. But you understand that this is used for measuring the transpiration. Okay, now we have come mostly to the end of the transpiration. So I am just going to give a recap. I told you that transpiration is the evaporation of the water through the leaves. And then we were discussing that most of the transpiration is through the stomatal transpiration, even though you get cuticular transpiration and the lenticular transpiration, and the loss of water in the form of the liquid is what is called as a guttation. Then we studied about the structure of the stomata. Then the different factors affecting the transpiration also we discussed. Now this I believe that this would have given you a very clear picture about the different aspects of a transpiration. And if you have got a doubt, I am just waiting for 1 or 2 minutes to raise the doubt. Please type if you have got any doubt in the message column. I am just waiting for 1 or 2 minutes. As for my part, I have finished the topic. Now you have to go and memorize certain things which are required for the transpiration. Okay students, bye. In the next class we will go to the next topic called transport of water. Bye.